Hello everyone, welcome to Barcode Biotechnology and welcome to the second session of our bootcamp, that is the preparation of the protein and ligand molecules for the docking purpose. So in the previous session, we have discussed about all these things, that is the drug discovery and the drug repurposing study. Okay, so in the previous session, what we have learned, that we have learned about the core objective of the computer-aided drug design. Then we have learned about the clinical trials, the process of clinical trial by taking an example of one drug of moxifloxacin. Then we have discussed about what are the different types of drug design, that is the structure-based and the ligand-based drug design. Then we have studied about the structure-based drug design and what are the various techniques to perform the structure-based drug design. Then in this session also we have covered the ligand-based drug design. So, for the ligand-based drug design, what are the major approaches we perform? That is the structural activity relationship. Then we have discussed about the most important one, that is the drug repurposing study. That simply means with having a single drug, can we have more than one property? As like I have mentioned in this session, that if we have aspirin as higher dose, it acts as an analgesic, anti-inflammatory, and antipyretic. But if we give that aspirin in a smaller dose, it acts as a blood thinner. Okay, that means when the cholesterol is deposited, the blood flowing get clotted or unable to blood flow properly. And aspirin small dose, it decreases the blood thickness and it acts as a blood thinner for the proper flow of the blood. Okay, so we also have discovered many things here in the drug repurposing analysis or studies. We have taken the example of these three drugs, that is the first amantadine, we have taken aspirin, we have taken, and these two drugs are also we have studied. So if you haven't watched this video, then just go through that and you will have a basic idea about the drug repurposing studies and what are the various process of drug repurposing, like structure based, ligand based, and a network based drug design or drug repurposing analysis. What are the workflow for the structure based drug repurposing and the ligand based drug repurposing? And then we have discussed about the representation format of any ligand molecules. So there are various formats for the representation, like PDB format is there, the protein data bank format, MOL2 format is there, HDF format is there, SMILES, that is the simple text representation format, and you can see this one, it is your JPG format or the pictorial format. Okay, apart from that, there is PDB QT format is there, but most of the cases for the preparation of the ligand molecule, we use these four formats or these three formats: the PD format, MOL2 format, or HTF format. Okay, so thank you for showing such love and some, like, you have seen all those videos. Okay, even in the first session, that is which is the session where we have discussed about the introduction, many students have been engaged, and that makes us to give you a very advanced level, from the basics to advanced level of the molecular docking course. Okay, so for that, in today's session, we are going to discuss about the preparation of the ligand molecule and the preparation of the protein molecule for the docking. So in this session, we will discuss about the preparation, and in the next session, we are going to perform the molecular docking. And all these things we are going to prepare or docking or visualization, it will be completely hands-on session. And by just clicking on that or by viewing this video and your laptop aside, you can also prepare the drug molecule, prepare the protein molecule alongside. Okay, so I'm not going to take much more your time, and let's start the session. Okay, so welcome to the session, and now let's start. Okay, so the first thing first, that is what are the different tools, the various tools that are used for the preparation of the receptor. So the very first tool and the most important one, that is the chimera or Inside the chimera, there is an executable file is there which is called as the AutoDock Vina. Okay, so by using the AutoDock Vina, we can perform two things. That is the preparation of the receptor and the docking process. Okay, so these two things have been done by using the AutoDock Vina. Here in the Discovery Studio, many of the students have known that this tool is for the purpose of visualization. Apart from that, it is also used for the preparation of the receptor molecule. Okay. Then for the preparation of the receptor, we have another tool that is called as the Pymol. And this Pyrex, it is for the preparation as well. But apart from that, it is the main and the only freeware version where we can perform the multi-ligand molecule docking. So this session is completely dedicated for the preparation of the receptor and the preparation of the ligand molecule. And the next session will be completely dedicated to different visualization and docking tools. Okay, so majorly we are going to discuss 4 to 5 docking tools. So don't miss that session and have focus on this session. Okay, so if you understand about how can we prepare the protein molecule, then you can clearly do the docking processes easily. Okay. So for that, we are taking the receptor molecule for today's session. That will be the dihydrofolate reductase, in short DHFR. Okay. This DHFR or the dihydrofolate reductase enzyme we are taking for the session, and this dihydrofolate reductase is basically, it is a bacterial enzyme. It is an bacterial enzyme that is obtained from S. aureus and as well as E. coli. Okay, so this dihydrofolate reductase is a bacterial enzyme which is obtained from the S. aureus or E. coli, and to inhibit that, we have to prepare some ligand molecules. So while preparing the ligand molecule, we'll discuss what is that drug molecule, what drug molecule we are going to use, and how can we prepare that drug molecule or the ligand molecule. So talking about the preparation of the ligand molecule, we can create such beautiful looking multicolored drug molecule by using these two tools, that is the ChemSketch and ChemDraw. And this is the most commonly using tool for the preparation of the drug compounds. Okay, so Pyrex is also used for the preparation of the ligand molecule, but for the optimization purpose, okay, optimization means, that is first thing, that is the energy minimization, and the second thing, addition of the polar hydrogens. We'll discuss briefly about all these things. So these two tools, that is the OpenBabel and the Avogadro, these two tools are very much important for your optimization. For the 2D structure generation, we can use the ChemSketch and the ChemDraw. Okay, so ChemDraw is also used at, it is having another property for the optimization. Okay, it is a very much important tool, because by using this ChemDraw tool, you can optimize your geometry of the ligand molecule. You can create such beautiful images. So you can see that this structure, it is the paracetamol, as you all know that paracetamol. Okay, there is the at the para position there is hydroxy group is there. So the name comes paracetamol, means end is OL. OL means hydroxy. Okay, and this one, it is an amide linkage, C double bond ONH. It is an amide linkage. So this structure you are visualizing, this one, it is the morphine. It is the structure of morphine. So in this structure you can see there is a bridge compound is there, and this is the dash bond. You know the dash and wedge. This type of bond is called as the wedge, and these solid bonds. like this. These are called as a dash. Okay, so let's start. We'll go sequentially. First, we have to prepare the receptor molecule for the docking purpose. And the docking purpose, the receptor will be take dihydrofolate reductase. Okay, so let's start it. I'll minimize these things. So now I have to create, before going to the preparation of the receptor, I have to create one folder. That one will be, let's go through here, select one folder, then name this folder that barcode, barcode biotech class. Okay, enter. So inside this, we have to give three new folders. Okay, the first folder will be your doc prep receptor. Okay, so this is one of your folder. Then the next folder will be doc prep ligand molecule folder or the drug prep ligand folder. And the third one, it will be your docking folder. Docking. Enter. And if you want, you can prepare another folder, that is the visualization folder. Okay. So in this way, you can organize your complete setup. So you can see that I have four folders. So the very first thing that I have to do, that I have to prepare my receptor. For the preparation of receptor, we use a tool that is called as to find out the receptor, we have to use one tool that is called as RCSB-PDB. Okay, RCSB-PDB. It is nothing but it is your protein data bank. It is the large set. Many millions of the proteins have been present in the RCSB-PDB data bank. It is called as a protein data bank. Okay, so in this data bank, you can find millions of protein and millions of protein structures, and if you want mutated structure, you can find also. Okay, so if you are working on a protein field and you are synthesizing proteins, peptides, you can deposit your work here also. After careful evaluation, your structure will be get deposited and a PDB ID will be generated. Okay. Now coming to our session, we have to work on dihydrofolate reductase. Okay. I had just have to go dihydrofolate reductase. Just type dihydrofolate reductase, and here you can see in the first one that's written as a dihydrofolate reductase. Simply click on that, and it will give you a name of series of compounds name. Okay, set of receptors name. So you can see that this first one, it is a Canadian albican dihydrofolate reductase. Next we have dihydrofolate reductase complex with methotrexate. We have Canadian albicans. So you can change your, sorry for this. So you can select or filter your receptor molecule by just going to this section, refinement, click on more, then you can see you have Homo sapiens, E. coli, then we have S. aureus in end also. S. aureus will be there. Okay, so S. aureus is the first. So just click on S. aureus and it will show you different S. aureus containing dihydrofolate reductase. Okay, so you can see that the S. aureus bacteria containing dihydrofolate reductase, that is a wild type S. aureus complex with ipasprime, S aureus complex to a TMP, that is a trimethoprim. Let's take this one because trimethoprim is also an antibacterial agent, and that will be very much easy for us to visualization. Okay. So now this is your interface. Okay. This is the PDB interface. Okay. So now let's go one by one that what are the things are given here. So many data have given. Okay. So the first thing first, it is the S aureus dihydrofolate reductase complex with the TMP. TMP is nothing but your trimethoprim, and the PDB DOI number means the DOI is given where? The DOI number is given to a research paper. Okay. If you go down, you can see that some of the researchers have done the study on the S aureus and dihydrofolate reductase, and they have submitted their work, and it is the DOI that is provided to the research work. Okay. If you click on the DOI, you will be redirected into their research work. Okay. You can see this. Then comes to the classification. It comes under the classification of the oxidoreductase. Okay. This enzyme, it is an oxidoreductase enzyme. Okay. And the organism, it is the Streptococcus aureus, and it is also found in the E. coli. And you can see it is written as the mutation. That is, mutation is there. Okay. So many enzymes have their mutation is there inside there. We can see that. Then going to the experimental section, this structure of the receptor, it is find out by using the X-ray crystallography. So many structures of the receptor or the enzymes are generally find out by using the process of X-ray diffraction or the nuclear magnetic resonance, that is the NMR spectroscopy. And here the resolution is 2 angstrom. Basically, what happens in the X-ray crystallography, the less the resolution, the more clear the structure is. Okay, okay. You can get it by taking an example of camera quality. More the pixel, more clear is the structure quality. Okay, here the two resolution, 2 angstrom. This is called as the angstrom. The 2 angstrom resolution have given this structure. Okay, then let's go to the second, the macromolecule section. I have discussed this section, the literature one. And now talking to the macromolecule, this is the dihydrofolate reductase molecule. It is having a single chain, and author it has given the name the single A chain, that is the X chain. The author have given X chain, and the sequence of this amino acid is 158 amino acids. Okay, I have already told you that the amino acids is the backbone of any protein or for any receptor. Okay, so in this structure, we are having 158 amino acid sequence, and this is the image here. And these are some of the things that are that comes under the bioinformatics. This is none of our thing. And you can see that in this chain A, with the author have described that trimethoprim is there, and apart from that, that is the NADPH. It is the source of ATP. As you know, in the comment section, kindly note down what is ATP and what is the full form of the ATP. Okay, so here the NADP represents a nicotinamide adenine dinucleotide phosphate. It is a source of the ATP. Okay, and the ligand molecule or the drug molecule, it is attached to the receptor, that is a trimethoprim. Trimethoprim, it is nothing but it is your co-crystallized ligand molecule. I will show you. Then these are some of the given things that are here. Experimental data by X-ray diffraction data, these are given. This is not needed for us. Okay. So you can see in the this section, the structure section, if I click on the structure, it will be redirected to the S audio structure. So you can see this one. It is the starting of amino acid sequence. The first amino acid that is present here, that is a tyrosine. If I select on tyrosine, the tyrosine will be pop up here. Let me make it reset the zoom and take it down. See, if I select tyrosine, the tyrosine will be start pop up. You can see the structure. The second one, it is the lysine, and as I have selected the lysine one is present here. It gets zoom in. So we can deselect this one, and you can see where are the lysine molecules are present, that is showing us in this chain. In this chain, wherever the lysines are present, it is showing like this. So we can just simply deselect this one and reset the zoom. And you can see this is the complete crystallographic structure of your S or S dihydrofolate reductase. And you can see these dotted things. So these dotted things are nothing but your water molecule. Okay. And at the end, you can see here this is the drug molecule that is attached. This one. This one, it is your drug molecule that have been attached, and another one that is the NADP, that is a nicotinamide adenine dinucleotide, which is attached here. Okay, if I click on the ligand molecule, you can see if I remove this drug receptor, see here polymer, I have just click on not to visualize the receptor, I have to only see the drug molecule and the ligand molecule that are attached to NADP and the trimethoprim. Okay, so this is the ligand molecule. Then I can simply remove the water molecule also. So this is your complete drug molecule that is present inside your receptor. Okay, so now the first thing, the polymeric carbon I remove, or I can present. Then the ligand molecule, it is showing in the ball stick format. Then the water molecules. So this is the complete setup of your complete receptor molecule. And here you can see that I have told you that CFRB, it is the PDB ID of the dihydrofolate reductase containing trimethoprim. Okay, so don't forget about that. So now the main thing, how can we download this structure? So you have here option of download files. You simply go to the legacy PDB format, that is the protein data bank format, and click on that. Then it will be started downloading. Okay. So now we just simply go to the download and got it, and we can paste it simply in our folder. That is, where is the barcode biotech? This one class. This will be our receptor. Okay. So now you can open it in the Discovery Studio and you can visualize these things. Okay. So the in the four tools for the preparation of the receptor, we have one tool, that is the Discovery Studio is there, AutoDock Vina is there, Pymol is there, and Pyrex is there. So let's start the preparation of the receptor by using the Discovery Studio. So for that, what you have to do, you simply need to open the downloaded structure in the Discovery Studio. As I have Discovery Studio, all the files which are in PDF format, that will automatically converted into the Discovery Studio format. Okay. So now you can see that my structure is open here, and you can just zoom in and zoom out. You can just rotate and see what are the amino acids, what are the two ligand molecules that are present. You can visualize the interaction. Okay. So now let me change the screen color. So to change the screen color, there's nothing to do. You just right click, then go to the color, go to the background and select in white. Okay. So it is very much useful for you. Okay. And it is very much look like a publication ready figure. Okay. So now what we have to do? We have to prepare a receptor. So for that, we have to go to the view. Then you can see here in the view that is one option is there that is called as the hierarchy. And the hotkey for hierarchy is Control plus H. Okay. So now as I have clicked on hierarchy, you can see there is an option toolbar is here given. Listen carefully because all these things are very much helpful and it will take some time to understand. So here you can see this is the 3FRB, the PDB ID, and inside that all these things are present. So the first X represents the receptor molecule. Okay, I just deselect this one. I have selected, then deselected. Okay, so the next X represents the ligand molecule. Okay, what are two ligand molecules are present? One is NADP and another one is a trimethoprim. You can visualize this like this. You can visualize the ligand molecules that are present. Here it is first ligand molecule. This one is the second ligand molecule. Then the third thing, it is the water molecules. The third X represents the water molecules, the dotted ones. Then the head atom. So the ligand molecules are also known as the head atoms or simply heteroatoms. Okay, so we have the head atoms here. Then the water molecule will be get selected if you click on water. Then click on the active side and it will show that where the ligand molecules are getting attached, which amino acids. Okay, so the yellow portion that is showing means these yellow portions of the receptor are binding to the trimethoprim and NADPS for the activity. Okay, for the drug activity, this amino acids of the receptor is participating. So what we have to do while preparing the receptor, you just have to simply click on the X, then the third one, which represents the water molecule, and go to this one that is the delete option and simply delete. Okay. So now you can see all the water molecules have been removed. So now the second thing what we have to do, we have to remove the co-crystallized ligand molecule that have been attached. So you simply click on the X that represents the ligand molecule or the co-crystallized ligand. Co-crystallized ligand means the ligand molecules or the drug molecules that are present before before the docking. That is the co-crystallized ligand molecule. Okay, and click on the delete. So now you can see you have the only single chain is left, only the single chain is left, and the active site and the ligand group has been already deleted. So this is your plain and this is your prepared receptor molecule. Okay. So now you can see that this is my receptor, plain receptor. Now here you can see there are two options are there. View interaction and define and edit the binding site. So we can just click on define and edit the binding site. Then we can go to here from receptor cavity from PDB site record. So according to the PDB site record, what is the active site or what is the main portion where the drug molecule may attach? That's the binding pocket, which I have already told you, the binding pocket thing. If you just click on from PDB record, a single spear-like structure is appear. You can see this spear. Okay, this spear is nothing but it represents that this is the area, the entire area that is covered with the red spear, not this ones, because this area has been covered with a single red spear. It is nothing but it is your active site where the drug molecule can bind. Okay, you can see that this area. So when we have to put the grid box by generating through the AutoDock Vina, we have to prepare the grid box and we have to select this area of the receptor most of the case to get the better activity or the better result or the binding affinity. So the binding affinity, as I have already told you, that the binding affinity is come in the negative score, and it shows that how the drug molecule is interacting with the receptor compound. Okay. In this way, by using the discovery studio, you can prepare your drug molecule or your receptor molecule. Okay. So this is the active site number one, and second, this is the active site number two. So we have two active sites. And if you simply see the active site number one is bigger because the NADP, if you remember the NADP, that is the nicotinamide adenine dinucleotide phosphate, that also present as a ligand molecule. But the most important molecule or the ligand molecule that is needed for us, which is an antibacterial agent, that is the trimethoprim. And the trimethoprim is located inside this area only. Okay, so this is tip site number two. So as we have two ligand molecules, it shows that two big spheres. One is site one and site two. But we have to need the active site number two because the trimethoprim is present at this area. So in this way, you can prepare your receptor molecule and you can visualize the area in which the drug compound may be present or the activity may be shown by just doing all this hierarchy. Go to the define and edit binding site. All these things you can do and you can prepare your receptor. So the receptor preparation is a very simple task. Okay. So now what you have to do, you just deselect this one or you can select this one, go to the file, go to save as, then go to the doc rep receptor, simply click on backspace and doc prep discovery studio. Doc prep receptor discovery studio. So and don't save, just press enter. Okay. You have to save it in the form of the PDB, protein data bank file. Okay, you can save in the PDB files because we have to prepare, we have to open this receptor in the uh, chimera and we have to perform the docking. Okay. So in this way, by using the discovery studio, you can prepare your receptor molecule. Okay. So the next thing, that is by using the PyMol. Okay. So I can close this one. Okay. So I have closed this session and now we'll prepare our receptor molecules by using another tool that is called the PyMol. So you can see here that is the PyMol is there. Just simply I have to click on this one. The receptor preparation is very much easy because we just have to delete the water molecule. We simply have to delete the co-crystallized ligand molecule. Nothing more and nothing less is there. Okay. So you can see this is the interface of the PyMol. So go to the file, go to open, then click on your folder and open the PDB. Okay. So you can see that your receptor molecule has been opened. So PyMol is basically nothing but a visualization software also, just like Discovery Studio. So you can see that two ligand molecules are present here. One is present here and another one is present here. So by the same process, you just go to the DHFP, this one. So there are five different sections are there. So the A here stands for the action. Then we have second, that is the S. S stands for the show. H. So here the S stands for the hide. L. L stands for the level, and the C here stands for the color. Okay, we can give the we can color our molecule like by the process of chain. Because it is a single chain, we just simply select any of this and it will color our receptor. Okay. If you have multiple chain, we can color by multiple chains. Okay. So based on that, we can here you can see that I have colored my compound. So now what the main thing we have to do for the preparation of the receptor is we have to go to the action, the first one. Then we have to go to here. You can see remove the hydrogen. Sorry, remove the water molecules. Okay. Below the hydrogen, we have to remove the water. Okay. So now I have removed the water. The all the dotted molecules you can see that have been gone. Then the next thing what you have to do, you have to go to the sorry, you have to go to the display and you can see that the first option is a sequence. Just click on sequence and you can see based on your color coding system, many of the amino acids have been aligned. So if you remember, we have opened our drug molecule in this PDB data bank. Okay. And when we click on the structure, we find out that this is our the first amino acid is the tyrosine. So you can see here that the first amino acid is the tyrosine and the sequence goes on. So you can see which amino acid, the tyrosine amino acid is present here in this blue section, deep blue section. The yellow section, it is starting from the phenylalanine, the 86th amino acid and this sequence goes on and as I have known we have known that the total number of sequence of amino acid is 158 amino acids are there. So we have to go to the pymol and go to the 157 58 these two. We just have to select these two, the NAP. We can see that NAP has been selected and we have to go to the edit or you can go to the sorry, I have some flu. We can simply go to the action and delete selection. Okay. Uh, So you have to select the NAP, then go to the action, then you can delete the section. For that, you have to go to here, hydrogen, remove the atom. Okay. So the atom has been removed. Okay. So this one is the NADP has been removed. And here the second one, that is a TOP. So TOP represents a trimethoprim. So for that, we also have to go to the action, then go to the remove atom, then it will remove the two amino acids. Apart from 156 amino acids, and two ligand molecules will now get removed. Okay. So in this way, we can prepare our protein molecule by using the pymol. Okay. It's very easy. What you have to do, you just have to remove the water, then you just have to remove the co-crystallized ligand molecule. Let's suppose you have downloaded any protein that does not have any co-crystallized ligand molecule, just only water molecules are present. So simply go to the pymol and remove the water, or you can go to the discovery studio, remove the water. Okay. So now we have to save this file. So go to the file, then the export structure, then go to the export molecule, click on save, and it will pop up a interface like this, doc prep receptor, and name it doc prep receptor, then pymol. Okay, then enter. Okay, so another thing I have to mention here, As like the Discovery Studio, we have to save our file in the PDB format. As you can see, I have saved this in receptor in the PDBX format. It is showing like this. If I select the PDB format, you can see it will be appear like this, the Discovery Studio format. And we can simply open and visualize this receptor in the Discovery Studio. Okay, let's change that the prep in PyMol. Okay, then enter. Okay, so this receptor has been saved in the form of PDB. Okay, I'm just saying what are the different tools that are using for the preparation of the receptor. Okay, so we have two tools we have covered. Now next, go to the another tool, which is your Chimera. It is for three things: visualization, your preparation of the receptor, preparation of the ligand molecules. Okay, so for that we have to go to the Chimera. Inside the Chimera, we have an executable file that is called as the AutoDock Vina. Okay. So if your Chimera is not having AutoDock Vina, you cannot perform the molecular docking. Okay. It's taking some time to open. It is initializing. So now this is the interface of Chimera, and whatever docking you have performed or whatever PDB you have opened, it will appear like this. So what you have to do, I hope that everyone is writing what I am saying. So the first step you have to go to the fetch. If you know the PDB code like this, we have know the PDB ID is 3FRB. So we have to go to Chimera and go to the fetch and it will open like this, and you just simply have to type your PDB ID. In our case, our PDB ID is 3FRB. So 3FRB, then enter. Okay. So now it is running and it's open the 3FRB. So you can see that this is your receptor molecule, and by using the chimera, it is very much useful because we can see visualize very properly by using the chimera. Okay. And also for the docking purpose also. So in Chimera, what you have to do? Okay, first thing, okay, go to the select, go to the chain. You can see it is a single chain as the author described and name it chain X. If I select, all the things will be get selected. So I can go to select again and clear selection. Okay. The next thing, we have to go to the chemistry. We can find out that there is carbon is present, hydrogen is present, nitrogen, oxygen. If you have to find out other ligand molecule like zinc, like calcium, sodium, so click on other and you can see that there are various elements name has been there. So if you want any element that is let's suppose you have to find the magnesium. You just click on magnesium. If the magnesium will present here in this receptor, that will pop up. Okay. So in our section, no magnesium is present in our, so we can remove this one. Then the next thing you have to go to the residue. So in the residue, we have two things. One is standard amino acids. As all of you know that we have 21 amino acid sequence. So if you want to go for a specific amino acid, let's suppose I have to search for the glutamine. Okay. I have to search for the glutamine. I select on glutamine. All the residue or from this receptor where the glutamine is present, it will be appear like this. Again, if I go for the residue and search for the glycine, so these are the key reasons where the glycines are present. Just simply place your cursor to at that site and you can see that it will show glycine. This one is glycine. And you can see at the glycine, the very right to the glycine, it is written 139. It is nothing but the 139th number of your chain is the glycine because we have 156 amino acids here and from 156 amino acid or the 139th amino acid is the glycine, and the X is your chain A. Okay. So what we have to here, we have to go to the residue. We select on all the non-standards, means apart from amino acid, whatever present in the residue will be called as the non-standards, and the standards are the amino acids. Okay, we click on all non-standard, it will select the water, it will select the NADPH, it will select the trimethoprim, and go to the action. Again, then we have to go to the atom slash bond and select on the delete. Okay, now your receptor is ready. But in this case, we have to add some more things. So what we have to do? So for that, we have to go to the tools, then go to the surface binding analysis, go to the dock prep. So now we have to prepare a receptor for the docking purpose. So what are the things that need to be done? We have to delete the solvent. So as we have already deleted the solvent, that is the water molecule is present. So just tick, or you can just remove this one. So solvent has been deleted. Then these are some of the default setting. We don't need to touch that. Then go to add hydrogen. So we have to add the hydrogen molecule. When you delete that receptor molecule, okay, when you delete the co-crystallized ligand molecule, at that time the polar hydrogen get deleted. So we have to add the hydrogen. We give some charge to the receptor. Then we have to save it in the mol2 format. But in my case, I don't want to save in mol2 format. I can save it in the PDF format instead. Then click on OK. So which type of hydrogen you want to add? Steric hydrogens or all the hydrogen? So steric hydrogens are not that much important for the docking because the steric hydrogens are only for the section where the double bond is present. Okay. If the double bond is present, they cannot move. But if we add all the types of hydrogen, polar hydrogens, to all this complete set of the receptors, that will be very much helpful for me to find out the pocket atom. Okay. So I just click on all consider hydrogen bond. Okay. Then I click on OK. Then you can see that the all the polar hydrogens have been added and it is representing in the white color. Okay. Then we have to assign the charge. So now here comes the thing. I have to teach you one thing, that is the in the section, that is the force field, that is the energy. Okay, energy optimization. This thing we have to learn. But now I just simply say there are three types of charge are there. One is your universal force field. Don't get confused. If you are getting confused, you can just pause and you can have some water, then rewind the video and you can again see the video. Okay. So first thing, there is a universal force field or universal energy. This charge is applied to the all the elements that are present in the periodic table. We have 118 elements in our periodic table, and this charge is applied, or this force field is applied to all the elements. Then we have general amber force field. Okay, so this particularly this general amber force field, it is applied to the proteins. To the proteins, we have to apply the amber force field. Okay, then the MMFF94, that is the Merck molecular force field. It is basically applied to the organic compounds, okay, like trimethoprim, like all the present ligand molecules, synthetic ligand molecule. We have to add the charge, that is the MMFF94, that is the Merck molecular force field. Also, if someone is having work on the phytochemicals like alkaloids, glycosides, tannins, resins, all these things, we have to add the charge like Merck molecular force field. Understand this one? The general amber force field. It is applied basically to the protein molecules and the MMFF94, that is the Merck molecular force field, applied to the organic compounds, the phytocompounds. If I can give you an example, like turmeric, okay, that is the curcuma longa. The biological name is curcuma longa, turmeric, and it contains what? Curcumin. Okay, turmeric contains the curcumin. So we can, if we are working on the curcumin, we have to give the charge to ligand molecule, that is the this MMFF94. We'll elaborately work or elaborately discuss about these charge things while we discuss about the preparation of the ligand molecule. Okay. So now for the preparation of the receptor molecule, we have to give the charge, that is amber force field. Instead, if we are having, instead of the receptor, if we are having the drug molecule or the ligand molecule, we have to give other charge like the MMFF charge. Okay. So it is dedicated to only receptor. We are not going to prepare the ligand molecule here. So that's why you can see there is no force field. That is the MMFF force field. Okay. Merck molecular force field is not here. Only amber charge is here. Okay. So now click on OK, and you can see in the left corner the duck prep has been finished. So you can identify here what will be our pocket atom. Okay, that will be also discussed briefly in the later session. So you can see that these things is showing the polar hydrogens. So we can prepare a grid box at this position at, to cover the drug molecule. Okay, we'll discuss briefly in the docking video, but currently just visualize this thing. So it may be our pocket atom because the drug can bind here because all this area has been covered with the polar hydrogen. Okay. So after all this thing you've done, you go to the file, save PDB, go to the desktop, then we have our barcode biotech class, docprep receptor, and name the file as docprep receptor, then the AB, that is the AutoDock Vina. So by using these three tools, we can prepare our receptor. If you ask me about all these three tools, which tool is very much accurate, I will say the AutoDock Vina, because in the all previous two tools, we just simply what we do, we do not add any charge. The charge is applied automatically. But in this case, we go add the hydrogens, add the charge by we do all these things by our side manually. So that makes give some satisfaction that our receptor is perfectly prepared. Okay. So now let's go to the preparation of the ligand molecules. If you are getting so much tired, then you can pause the video, drink some water, then we'll go for the tools for the preparation of the ligand molecule. Okay. So I have already told you that ChemSketch and ChemDraw are there. Pyrex is there, OpenBabel and Avogadro for the optimization. For the preparation of a 2D structure to 3D structure conversion, we need ChemSketch and ChemDraw. Okay. So let's have some idea about the force field, which we are going to apply in many sections, like for the preparation of the ligand molecule, for the preparation of the receptor molecule. Okay. So what is force field here? Okay. So talking about force field, it is nothing but the potential energy of the molecule. Okay. So what is potential energy? Sorry. So there are two types of charge. One is your potential energy. two types of energy. Force field is nothing but you can say it is an energy of the compound, okay, or energy of the receptor. So there are two types of energy. One is the potential energy, one another one is the kinetic energy. So the potential energy, it is the energy that is present in our body. Just like the just like us, the energy that has been reserved in the ligand molecule or the drug molecule. Okay, that is called as the potential energy. That is our own potential. If anyone is giving us, borrowing us some power, and we are having own power, and someone is giving us power, that is the kinetic energy. Kinetic energy means we are getting energy from outside. Okay, so let's consider one thing. Let's suppose this one is the benzene. Okay, so the this benzene is reactive in nature. Okay, it is reactive in nature. But when in this benzene ring, I add one nitro group, which is an electron withdrawing group. This makes the benzene more reactive. So by this example, you can understand that this benzene, it is having its own potential energy for the reaction. But when I add the nitro group, which is an electron withdrawing group, it will pull the electron cloud toward itself and make the benzene more reactive. So that is the kinetic energy, and this benzene is purely this the potential energy. Okay, so this is the difference between the potential energy and kinetic energy. So here the force field in the drug ligand preparation describes the potential energy of a molecule system. If we have any compound, then what is the potential energy of that compound? That is called as the force field, or simply energy of that compound. So how we calculate the force field or how we calculate the potential energy? So it is basically calculated by two terms. That is, one is the bonded and non-bonded. Okay, let's consider one thing. Okay, so now we have a structure of benzene. So what are the parameters of this benzene ring? What are the things we can visualize? So we can visualize the bond length. This one, the bond length. So the bonded thing is the bond length, LENGTH. Then between these two bonds, what is the angle is creating? So that will be the next thing, that is the bond angle. The third thing, that will be the dihedrals. Okay, so dihedral, it is nothing but it is the vibration or rotation. Okay, what is the rotation? That is the flexibility. So these three terms, that is the bond length, bond angle, and the torsion or flexibility, these are the bonded terms. Okay, these are the bonded terms. Now talking about non-bonded terms, so non-bonded terms are nothing but the interaction of the receptor towards the drug molecule. So now take an example of this benzene only, and this benzene, and here it is the NH2 group of the amino acid is there. Okay, so the amino acids generally contain one NH2 and one carboxylic acid group, one amino and carboxylic acid group. So this carbon, okay, this carbon and this nitrogen is some of the connection will be there if if we perform the docking, and there might be chance the carbon can react with this NH2 group and it will make some bond or attraction. So what are the different type of attraction here? You can see that the van der Waals interaction may be possible, or electrostatic interaction may be possible, or hydrogen bond may be possible, pi-pi interaction may be possible, pi alkyl interaction may be possible. So these are the two terms, that is the bonded term and the non-bonded term. Okay. By calculating all these things in mathematical calculation that happens in the back end, okay, By just simply calculating all these things, we can say that this is the force field. So what are the different type of force field? As I have already mentioned, the first one, it is the universal force field applied to the periodic table elements. Okay, all the elements that is present in the periodic table. In that case, the universal force field is applied. Then the second one, it is a GAFF, that is the general amber force field. It is applied to the protein molecule. Then the third one, MMFF94, that is the Merck molecular force field. It is applied to the compounds, organic compounds and the phytocompounds. Okay, so if I have any phytoconstituents, in that the Merck molecular force field is applied. Okay, so now talking about the ligand energy and optimization. So the total energy will be E bonded plus E non-bonded. So, so total energy or the total potential energy of the compound of the force field is equal to bonded plus non-bonded. So bonded continues the bond length, bond angle, torsion, while the non-bonded are the electrostatic interaction, van der Waals interaction, all these things. So it is generally used in computational chemistry and molecular mechanics to describe the potential energy, of a molecular system. Then this is the important point. It represents the sum of all the force or energy that hold a molecule together and dissects its interaction. So, in very simple language, it is the potential energy of the compound, and we have to stabilize all these things. In the next slide, it will be crystal clear. Okay, so now see, if I draw a compound or a ligand molecule like this by using the chem draw or chem sketch. Okay, so now this compound, you can see this is the benzene ring. Let's assume one thing. This is the bond length. This is the bond length. This one is the bond length. Okay. So let's assume the bond length of these two carbon between the bond length is 2 angstrom. This one is a 3 angstrom. This carbon and this carbon bond length 3 angstrom. This one may be 2.5 angstrom. Okay. This carbon and this carbon having the bond length of, let's suppose, 4 angstrom, and it will be very much high. Let's suppose it will be 5 angstrom, and this one, let's suppose it is 2 angstrom. And talking about the bond angle, let's think that it is 120 degree. This one is other like 130 degree, like this. Okay, if you can see here, all the bond length and all the bond angle, not they are not uniform. Okay, so when we apply the force field, so as it is an organic compound, when we apply the force field, that is the MMFF94, that is a Merck molecular force field, it will convert this compound into a compound, simple happy phase compound, where the bond length will be 2 angstrom for each two carbons. Means this carbon and this carbon is having the bond length of 2 angstrom. This is same for all. Okay, I will show you an example by using the Avogadro. Don't worry about that. Means all the compounds is having the same bond length. And talking about the bond angle, all the compounds, these two carbon is having the same bond angle, and these two is having same bond angle, and these two is having same bond angle. Also this one also. So it implies that we make it into the equilibrium state. So as I am saying, the E bond length is the at the equilibrium state. When the bonds start to vibrate, what is the bond length? Okay, take it as an example. Okay. So here we have the benzene. We apply some radiation towards it. Suppose we apply the IR radiation on it. Okay. When we apply the IR radiation, what happens? The molecules start to vibrate and we get a graph like this, stretching, bending, all these things. Okay. So when we apply the IR radiation, when the molecules started to vibrate, at that time, what is the bond length? Okay, that is the equilibrium bond length. You can see this point oscillation about the bond length at the equilibrium state. When the compound is in equilibrium state and we apply any some sort of energy like NMR radiation, IR radiation, at that time, what is the bond length? That will be called as the E bond length. Again, the same thing about the bond angle, oscillation of the three atoms at the equilibrium bond length state. Okay, when the compound exposed to any radiation and it is having the bond length, at that time, what is the bond angle between this carbon, this hydrogen, and other things? Okay, so what is the bond? That will be the equilibrium bond angle. And about a torsion, at that time, how it is getting vibrated. Okay, so torsion rotation of the four atom about a central atom is called as E torsion. I hope you guys understand all these things because it is very much necessary to understand the force field for the preparation of the ligand molecule. Okay, let me show you an example. Let's keep it here. Oh, I can close this one. So now what we have to do, we have to open the chem for the preparation of the ligand molecule. Okay, so this is the interface of ChemDraw, and these are some of the options are there. It is file, so you can open any file here, the drug molecule file. Here is the option of edit. So when you go to the structure, you can convert the name to structure. Okay. So now let's name one structure that is the trimethoprim, which we have taken as in the receptor. Trimethoprim. So when you click on enter, it will open the structure of trimethoprim. You can see. So this is the structure of trimethoprim. Okay. So now you can see this is the pyrimidine core is present. This one is the pyrimidine core. If you want to give some color, then you can just select that portion and you can go here and you can give some color. But here in case we don't need any color because we just have to prepare our ligand molecule for the docking. So for that, what you have to do, we have to select the whole compound. Now we have to go to the view. You can see here that there is an option called a Show Chem 3D Hot Link window. Just click on Chem 3D and you can see the 3D structure of the compound have been appeared. So we have to launch the Chem 3D. Click on the launch Chem 3D and it will open. See. So now you can see that this is the 3D structure of the compound and you can see that this is the bond length is given 1.34. This is 1.34. So the structure is optimized. But to validate that, we have to again optimize the structure. So for that, what we have to do? We have to go to the calculation. Okay. So we have to go to the calculation. We go to the Merck molecular MM2, then minimize the energy. Okay. What happened? When we minimize the energy, the structure became it comes into its equilibrium state. Just click on minimize energy and these are some of the default settings. We don't need to do any changes and click on run and you can see that the structure is moving. So and the energy minimization is going on. Okay, you can see that the structure is getting little bit modified. So It will take some time, like approximation 30 to 40 seconds, or sometimes it takes 1 minute, and you can see that the energy minimization is going on. It is minimizing the energy here, and you can see the total energy at the very end. It is a 27.9 kilocalorie per mole, and it is the 3D representation, 3D visual representation in the plane. Okay, 3D plane. Okay, this is the final structure by using by minimizing the energy, and by doing this you can see here it is changing. Okay, the 2D structure is changing. Okay, so see, the benzene ring is present here, but in 2D you see this pyrimidine moiety is present aligning to the benzene, means they are present in the same plane. But here you can see the pyrimidine moiety. Okay, the pyrimidine moiety is present backward. See, this is your benzene and the pyridine is present below or backward. Okay, so this is the 3D representation, and this one it is a 2D representation, and that is the difference between the Chem 3D and the Chem 2D. Okay, and for the docking purpose, we always need the 3D structure and the optimized structure of the ligand molecule. Okay, so now what you have to do, we have to go to the file, then go to the save copy, then go to the desktop, then to then go to our barcode biotic class, then go to doc prep ligand molecule and name it as ligand 1 TMP trimethoprim, then 3D. Okay, and the format I have already defaulted the PDB format. You can change, but you don't need to change. You have to save it in the PDB protein database format and then save. Okay, so you can see the ligand 1 TMP is not came 3D format. Some information might lost. Save. That is unnecessary because we have our energy minimization. All these things have been done by using the CAM 3D. Okay, let me show you another tool and we'll wrap up the session. After that, you can just simply select no and done. Okay. So now the next software, that is the OpenBabel. Okay. So for the OpenBabel, what you have to do, let's go to search OpenBabel. Open it. So it will take approximation 2 to 5 minutes only. Okay. So what will be our input format? Okay. It may be HDF or it may be many software. There are guys, we have having many formats for the input, but we select the HDF format to see. So to introduce the HDF format, first we have to go to the pubchem, this one pubchem, not pubchem feature, the pubchem only. Okay. So now click on pubchem and click on it, write your desired compound name. I have already tell you about how to search in pubchem, that the structure name is trimethoprim. Then you can see trimethoprim, and this is the trimethoprim structure and the information that has been given here. Okay, let me recall all these things to you. So this is the 2D representation, the 3D representation, and the X-ray crystallographic structure, and these are the lab grade things. That what is the property? It is irritant in nature. It is health hazardous, environmentally hazardous also. And these are the synonyms and the CAS number for the industrial purpose. The molecular weight is given, that is the 290.32. And what we have to do, we have to download the 3D HDF format. So go to the download and download the go to the 3D conformer and download the HDF file. Okay, so now it is have been downloaded. So you can see that the Discovery Studio format, if we download in the PDF format, it is showing the Discovery Studio format, but it is shown like HDF format, so it is not in Discovery Studio format. Okay, so now what we have to do? We have to go to the Open Babel, then we click on this three dot line. That is, we have to upload, go to the conformer, it is go to download and go to the conformer. Okay, so now it is open. Okay, so in this section, what you have to do, first of all, you have to add the hydrogen. As I have mentioned that to prepare a ligand molecule, we have to add the hydrogen first. Then you have to generate the 3D coordinates. You can see you go one by one and you can see that here it is generate the 3D coordinates. Then you go to the plugin, then go to the force field and that is the MMFF94 force field. Apply the force field, then convert. You simply click on convert and it will be converted. Okay. This problem happened because I haven't give the output location. Okay. So no worries. We'll try again. First, the HDF format we have to take HDF format. Then go to the input file, click on it, enter. Then we have to add the hydrogen, add hydrogens to polar only, polar hydrogen because we need only polar hydrogens. Then click on the 3D coordinates. So click on generate the 3D coordinate. Then go to the plugin force field. The MM94 force field is applied. Okay. So now the, for that you have to select the output file location. Okay. So the output file location will be go to the desktop, then go to our class doc prep ligand, name the file that is the ligand one using the 3D ligand one 3D that is the open babel OB. Okay, then save. So you have our output file location and then click on convert. Okay, so now it will convert the structure. Okay, so there is some error is showing, so that's why I think it is closing automatically. No worries about that, but we because we have already prepared the drug molecule, so you can see that the open babel ligand molecule has been prepared. I just open it in the PDF format. Okay, okay. So there is some error is showing. We can prepare the drug molecule using the open babel in the next session. I will show you the Avogadro only. Okay, so we have discovered about the ChemDraw and we now just have to discover these two tools and the session is very lengthy right now. So we have to wrap up the session and then the next session I will show you the preparation or optimization of the ligand molecule using the Avogadro and the Open Babel. Then we will go for the blocking only, okay? Because it is already being 1.5 hour session. Okay, so no worries about that. We'll learn all the things. It will take 3 to 4 sessions, but we'll complete all the things step by step. Okay, so don't worry. And thank you so much for joining this session and taking the session this much longer, watching the session this much longer. It takes some patience. Okay. So no worries about that. Thank you so much for joining and see you in the next session. And in the session, we will cover these two tools, the Avogadro and the OpenBabel, and we will perform the docking using the Chimera also. So thank you so much for joining the session. Thank you so much.